G'day, g'day! Hey guys, sorry I couldn't make it over for your meeting, but I thought I'd bang together a quick video to explain what I've been up to. Thomas was quite keen for me to explain what I've been doing with all those dead stoats that you sent me in the mail. So, for those of you who don't know me, I'm Jamie. So, some of you have been involved with this project of mine in the alpine zone of 4 different national parks, and you guys were collecting dead rotten stoats from the traps in the Murchison Mountains and in the Hollyford faces and sending them off to me in the mail, yum yum yum. And I was doing some study here at Otago University to try and figure out what stoats are eating in the alpine zone and try and figure out what's getting eaten by stoats in the alpine zone. So this is part of like a bigger piece of work, which is trying to figure out how we better protect alpine species, if we can figure out the differences between what stoats are eating in different areas or different times, and perhaps we can figure where our native species are at greater risk or when they're at greater risk, and we can choose kind of where or when to put our limited money or resource in a time or a place where it's going to have the maximum impact. So first of all, thank you guys heaps. Thank you so much for sending me all of those stoats. I really, really appreciate it. I understand it's like totally not a small thing to do to ask someone to, on a multi-day trapping trip, take a rotten, stinky, maggoty stoat and put it in a plastic bag and put it in their pack. So I really, really appreciate what you guys did. That's a huge help for me, and it's really awesome for conservation to have a group of trappers like yourself that is just so much thoroughly behind what we're doing in terms of trying to do what we do even better, and that science of pest control and improving how we're protecting these species. So awesome. I super appreciate it. Thanks heaps. So what I did is I collected stoats from your guys' sites, so the Murchison Mountains and the Hollyford faces and all those traps around the tops and the root burn. As well as from Kahurangi National Park around Mount Arthur, and the Friends of Cobb and the Friends of Flora have got some trapping operations in the tops up there, and also the St Arnaud Range in Nelson Lakes National Park. So that's another network of traps that's all along the St Arnaud Range, sort of up by Rainbow Skifield up there. They got all of their stoats as well. So all of these stoats just caught in the alpine zone. And what I wanted to know is, what were the stoats eating? And what was sort of the factors that maybe affected what they ate in different areas or different time frames. And there's been a little bit of work done before. A guy called Des Smith did some really cool stuff in the Murchies in the past and in Borland, but it's always just been one site or one season, and the ability to look across more time frames and more different sites means we've got way more predictive power in terms of being able to say, oh, well, what's affecting that and what's different between those sites and what might be different. So I got all those dead stoats, yummo, super yum, pretty maggoty most of them, as you guys know, and I cut them up and looked at what's in their guts.
What do you got there, man?
It's a particularly disgusting stoat. Oh, there's a big name there.
Classic gut content analysis, snip them open and record the percentages of different things in their guts. And so at your guys' site, so Fiordland was mainly I think every single stoat I got from Fiordland had mammals in the guts, so that's likely to be rats, mice, and maybe not in Fiordland, but other sites there's some hares in there as well. The Mount Aspiring site, so that's the root burn tops area, was a combination of things. Still a lot of mammals, a bit of invert. And then at the Nelson Lakes National Park site, I found a really big variety of things. A lot of lizards, a lot of birds, a lot of mammals, bits and pieces of plants, sort of really big variety of things. And the fourth site, Kahurangi National Park, Graham Elliott's team came and 1080'd the whole site, and I got very little indeed. But great for the rock crane and stuff up there, but I dropped that site from one of my study sites. So that's really cool and that's really useful, but it sort of only tells us what they ate in the last 4-6 hours. Stoats have got this super fast metabolism, so they just chew through food. They've got to eat really regularly, so it's kind of helpful to know what they ate in the last 4 hours, but it's nice to be able to get a lot more information, and that's where I used this kind of biochemical stuff I was doing. So I took this tool that they use quite often in archaeology stuff, and sometimes with marine mammals they do it a lot to say like, oh, what do whales eat, what do dolphins eat, that kind of thing. And essentially it involves looking at the chemical signature of all the different prey items, and then looking at the chemical signature of the consumer, of our stoat, and depending on what the chemical signature of the stoat aligns with, it'll tell us what the different prey items sort of were in the diet, and so you do some fancy mathematicsy, statsy stuff to say how much of the different parts of the diet were made up from different prey items. So I took them all to the lab, and I used some claw collagen, so little piece of like sort of fingernail from the stoats of their claws, and some bone collagen as well. So the benefit of those two is that even in the stinky, ruddy state that some of those stoats were in, obviously the fresh tissues have degraded and things like that, but the those longer tissues, bone and claw, just stay like they are. So it means I could sort of access that for a much longer period of time. And the chemical signature of those tissues is made up of, is kind of turned over at a much slower speed. So you might have heard people say, oh, you completely replace your skin every 7 years or something, and that's to do with just the turnover rate in the cells. And so bone collagen is a very slow turnover tissue, so it probably represents the lifetime of these stoats. So the chemical composition of the bone collagen will tell me what they've been eating over their whole lifetime, whereas the claw, like your fingernails, stoat claws just grow continuously at a slow rate. So I took a millimetre section just closest to the skin of the claw, and that tells me what the stoats have been eating for about a month, I think. That's sort of estimating from some growth rates from different other species, but probably about a month worth of diet is telling me what they're eating. I also used the liver of some of the stoats that were more fresh, some of the really nice fresh ones. I used the liver, which tell me that, but that had quite a small sample size because most of them were pretty ratty. And so went into the chemistry lab and did all sorts of chemistry tricks and test tubes and hydrochloric acid and all sorts of fun stuff. Extracted the collagen, took the claw, and I put them all through this fancy machine called a mass spectrometer, which is a mass spec, essentially weighs the isotopic weight of different chemicals. So in this one I used carbon and nitrogen in them, and gives me that chemical signature, and allows me to look at what they've been eating. And long story short, across the different areas they were eating a huge variety of different things. Like the stomach contents, I can see that in Fjordan National Park, hugely reliant on small mammals, whereas Mount Aspiring National Park was a combination of sort of mammals and invertebrates, insects, that kind of thing. In Nelson Lakes National Park, just like the stomach contents, it showed that they were eating a really big variety of things, so a really big range of different food items. So it sort of backs up what the stomach contents said, which is good, kind of, hey, this chemistry stuff might work all right. But it also, it's a really powerful tool to be able to see what they're eating. And so this is a really nice tool to be able to see over the long term what they're eating. And because I had those 3 time periods, I can compare one time period to another. So for example, with the claw tissue, the recent tissue, it looks like they had a stronger reliance on insects, things like weta. So all of those big ground weta that are up in the Murchison Mountains, they were eating those more in summer when it's warmer and those weta are out and about more. So that's kind of one theory we had, that the weta were probably overrepresented in the diet of stomach contents because we're always doing that in summer, and that actually that doesn't make it. The long term we look at the bone collagen, and it's long term it is the mammals that are really important. And so then I got to looking at, well, why? What are the things that are affecting what stoats are eating in those different areas? And I ran a model with a whole lot of different predictive variables, trying to tell me, wow, which of all of these different things, which one is important in telling me what stoats were eating. And so I put in there like sex, age of the stoat, the elevation it was caught at, the site it was caught at, all sorts of different things, including the number of rats that were present on the ground, and also the number of stoats that were present on the ground. So do they eat lots of rats when there are lots of rats there? Or do they eat lots of other things when there's perhaps lots of stoats there and there's lots of competition for food? And those two things came out with pretty much the only things that came out as kind of significant predictors in the model of what they're eating were those two things there. So the stoat competition to a lesser extent, a smaller driver, but certainly the the number of rats around, so the trap catch of rats in the traps, had a really strong influence on what they were eating. And it seems like when there are lots of rats around, they're a big, juicy mammal, like very energetically rewarding for stoats to hunt rats. The stoats pretty much just ate rats. Whereas at Nelson Lakes National Park, where there are no rats, or there were no rats in the season I was up there, so they caught 0 rats in all of their traps all season, and that's when stoats seem to sort of branch out and start relying on lots of different prey items. That's got some really interesting consequences for how we manage things. So, for example, you guys know that sometimes Doc will deploy extra trap checks in the Murchies at the decline phase of a beachmasting rat plague. So when the rat numbers start to drop off, they perceive the takahe as being at greater risk. So this is a really good example of some sort of empirical evidence of how that's happening. So it might be that that as the rats become less available in the diet, the stoats start preying on other items, and that dietary niche, the range of food items they're eating, gets bigger, and our things like takahe are at greater risk. So the hope from here is that we can take the information that I've got from this study, things like that, things on those periods and times and areas when there is greater risk, and we can use that to inform sort of some trapping best practice and how we're managing the alpine zone, how we're selecting sites and times to manage, so that we can make sure that we're actually just putting our trapping effort in the time or in the place where it's going to have maximum impact, where it's really going to hit home and save stuff. And perhaps there are other times when our native species are at lesser risk and we can scale back that effort and put it somewhere else. So yeah, hopefully that gives you guys a good idea of what the whole point of this thing is, like why I asked you to put dead stoats in your pack and send them off in the mail. I really rate what you guys do every day. Like, people like you who are out there trapping are really doing the mahi and saving stuff, and so it's the main motivator and driver for me doing this study was to make your guys' job easier, and I really, really appreciate your help in sending me stoats and being part of the study and just being so eager and keen to be involved. So thanks very much, and hopefully this kind of helps and gives you some context as to what it's all for and what it's all used for. And yeah, we might see some of you for a beer sometime in Te Anau. Sounds good. Cheers.